Well, let me tell you that it is my pleasure to introduce our next speaker, Professor Gerd Binnig. We're truly grateful that he could make it to Clinam. It's an honor to have him here. As you know, Professor Binnig is well known everywhere for the discovery of the scanning tunneling microscope. He joined IBM research facilities in which he co-invented the tunneling microscope together with his colleague, Professor Heinrich Röher. After that, and because of this, they both received the Nobel Prize, as you know. Professor Binnig has received a lot of different prizes along his brilliant career. One additional invention from him is also the atomic force microscopy, which is a tool that most of you use in very frequently basis. So, and now I know that he has recently endeavored a new challenge, which is a company called Definiens. Need my glasses on. Yeah, a company called Definiens. Yeah, that's right. Thanks. Which is leading with some specific software to analyze a very large amount of data, especially from tissue imaging. I'm positive that this will be of great useful for many of us, especially in the medical field. And this has actually opened the field for a new definition of image analysis, which is called tissue phenomics. And actually, I think that you will be talking mainly about this tissue phenomics in your talk. So, Professor, thanks very much again for your coming here in to Clinam, and really good to hear you. Thanks.
Yeah. Thank you very much for the kind introduction and the nice words. It's always a pleasure to be here at the conference. It's not the first time I'm here, and I always enjoyed, and the discussions with you were usually very great. Today I will talk about, in a way, how to create knowledge automatically, which is something relatively new. I mean, usually you create knowledge by thinking about something and by doing some experiments or by discussing with other people. And then you can have something to think about again. So having a network of people is always very helpful to create knowledge. And that's why we are all here. And technology always helped in this respect, that people can communicate with each other, that they can meet each other. It goes back to centuries. Like the first really important invention was probably printing, where you can read what other people think. And by this you get stimulated, and maybe you create also this way new knowledge. But there are many different inventions over the last centuries in this respect. But it's mainly helping to create knowledge. It's not creating knowledge automatically. The computer changed that a little, because on the computer itself you can create knowledge in a way automatically. For instance, if you do a simulation of a complex molecule, how it gets stable, you just shuffle it around, all the atoms, and it by itself find a stable configuration. In a way, the computer invented a new molecule. It can happen, yeah. But on the other hand, you put in already a model from quantum mechanics, for instance, yeah. You put in a model, a very specific model, and you start with that. With big data, it's a little different. You create knowledge, but not putting in a model in the beginning, or you put in some algorithms, very generic models, but not a specific model. And that's very new. So the machine creates knowledge more or less by itself, by just collecting data. It's a different way of creating knowledge, because it's the old-fashioned, I would call it the old-fashioned way of creating knowledge is by cause and effect. You think about what could happen. Things interact with each other, and as a cause or as an effect, something is happening. And then you understand the whole network of interactions that take place. So that's a way how we usually create knowledge. With a big data approach, where you just do data mining and you just look for correlations between certain events, that's slightly different. You don't think about cause and effect. You just look at correlations. But once you've found correlations, you can go back and think, what actually is here the cause and what is the effect? So the both types of research can influence each other very much. And that's happening now in the clinics a lot. Big companies like Google or IBM collect data, all kinds of data in the clinic, and that's really big data. And correlate, for instance, with clinical outcome. You can do this. We focus at Definiens on a small section of that. We concentrate on tissue. Tissue is very important for cancer diagnosis, and in principle for cancer at all. It plays a very important role where you can see what's actually going on the microscopic level. And it has something here other methods or other data do not provide. So you can look at the cells. You can look at the state and the type of a cell. You can look at the cell interactions. Do they do something with each other? Are they close or far apart from each other? And you can look at the arrangement of all the cells in your tissue slide. And that's something other methods cannot deliver. I would hope that in the future, maybe in radiology, some invention is made where you can look at cells and how they interact, for instance, the immune cells, how they interact with a tumor in MRI or whatever kind of. This does not exist yet, but it would be wonderful if nanotechnology would come up here with a solution. Yeah, this is very important, mainly for situations where you want to look at a kind of a battlefield. For instance, if you want to study the interaction of the immune cells with the tumor, where we are most interested in, then it's kind of a battlefield. You want to see how they interact. And then you need exactly this information other methods don't provide. There's not always only advantages. Sometimes you have also disadvantages. And one disadvantage of this method is if you collect a tiny little piece of tissue. After surgery, it could be a bigger one. It's a resection. But if you just do a biopsy from the lung, for instance, it's sometimes only a tiny little piece of tissue. Can you learn something or predict something if you just look or quantify such a tiny little piece of tissue? That's a question. And we looked into that and tried to find out whether you really can predict something. The great benefit is if you have this piece of tissue, you slice it very thin, and then you can stain it. You can stain it that you see all the objects of interest, like the cells or the nuclei, but you can stain certain proteins. This is also a very old type of nanotechnology, making tiny little objects like proteins visible through staining, enhancing the contrast by attaching certain molecules to certain proteins. A very old invention, but in a way it was already a nanotechnology. And you can also stain the genes, same result. And when you now want to do big data on those kinds of slides, then you, in principle, can suck in all the information you get from those slides, put it into a big data machine, into a data mining machine, and this machine has several algorithms that can be applied, and it just looks for correlations within those data. In principle, this works like this, but you need some structured data. If you just put in meaningless data like pixels from your images, it does not make any sense. You just spoil your data mining machine. So you have to structure the data first. You look just at objects, and maybe even objects like cells is not so important because the meaning of a single cell is not that great. Usually you have millions of cells in such a tissue slide, and you need to do some statistics about those cells. That tells you much more than just a single cell. So structuring even more into regions where you want to do the statistics is very important. And the process how to now create this knowledge automatically is very similar to the process that already occurs in the clinic, in the daily routine when people, for instance, get diagnosed with cancer, for instance, or other diseases. So in principle, a piece of tissue is taken, it's sliced, cutting it into very thin slices that you can look with an optical microscope onto the slide. It gets stained, like I said before. Proteins, in most cases, IHC, so it's the proteins get stained. And then it gets scanned. That's now different. Not all the slides that are produced today get scanned. Most of them just get looked at by the pathologist, and he looks at all the different cells he can look at. But it's coming more and more that people scan it, and that you can look on a computer screen then, and do what comes later, the image analysis on those slides and quantify what you get out of it. So you create data and not just looking at it. And then once you have those data of all the cells you collected or measured, then you put it in the data mining tool. That's how it works. And you predict this way an algorithm that tells you about the correlation. So whenever some pattern like this occurs, then there's a good prognosis or it's a bad prognosis. That's a prediction. And maybe a more important prediction is if a certain pattern occurs, some drug might work very well for this patient and another drug less. So predicting drug response is maybe something that's very important here. So and once you discover that and you bring this algorithm into the clinic, then the process is very similar. You do the same thing, but you don't need any data mining tool anymore because you already discovered your algorithm. You just apply it to an individual patient and just say, okay, this patient is good for this and this treatment and not so good for other treatments. So the first step is you do image analysis, and in image analysis, a pathologist knows that in many cases he uses simply context, not just looking at the cells. If it just focuses on a single cell, he will, in many cases, will not be able to tell you what kind of cell it is. So you sometimes have to look at the context in the neighborhood to understand really the full picture. So we are using context-driven image analysis, and at the same time, you not only look at the neighborhood of a cell to come to a better conclusion, but you also do the processing over time. So you first look at the more simple objects, and from this you create some context. So to find the more complex object. It's because those images are not so easy to analyze. Actually, sometimes you have lots of artifacts. You have some stains that did not work, or the cutting was created stripes or whatever, and many different artifacts can occur, and you still have to analyze those images in a very reliable way. And that's why we do also over time do the image analysis stepwise. So, for instance, we look first for the very simple objects. Sometimes you get clusters of cells and clusters of nuclei or cells, yeah, and they are not so easy if they are, for instance, cancer cells. then they have a strange shape and very heterogeneous. And if they touch each other, then it gets even more complex. So what you can do, you can look just for the cells that are not touching each other, isolated cells. You find them by relatively simple methods, and then you train by artificial intelligence to find those cells. Because now you can train automatically without having looked at the slide beforehand. You train that, and then in the next step, you can look for similar cells in the whole image, and then you find also the ones which are touching each other, and so the more complex ones. So context-driven image analysis is something that is here very helpful and very important. And that's something like a result you can see here. You apply the same algorithm to very different kind of images because it's self-trained. It adapts to the image this way, as I explained, and this way you can actually provide the algorithm is very different type of images, and still it recognizes, for instance, here the nuclei, as you can see, or here in this case a different stained image, same algorithm, get same similar nice results. So once you have now found all these structures that are relevant and important to you, you can do two different things. You just can imitate the pathologist, so automate what the pathologist does. That's one thing. The other thing is you can create new knowledge by looking for these correlations, so going beyond the pathologist. The pathologist already knows a lot, and he has seen so many different patterns and can we relate it to certain diseases. He's doing an excellent job here. But you might be able, by quantifying all this, to go even further and discover structures the pathologist has never discovered before. And that's then done through big data or data mining. In the first case, where you just imitate, the pathologist tells the machine what to look for. Usually it does it by annotating the objects that are of interest. Here's a tumor, here's a cell, and now the machine should find it. But if you do go beyond the pathologist, if you want to discover something really new, then the ground truth is not the annotation of the pathologist, then the ground truth is the clinical data, the outcome. So the prognosis, or what happened to the patient later, that's your ground truth. And then you try to correlate that to the patterns you observe. Yeah, we focus mainly on tumors right now. I'm a small company still, and you have to focus on something. So it's tumor, but the tumor itself is already quite complex. So that's what we see, because we stain the different slides differently, and we look at different proteins. And then you can actually realize it's really heterogeneous. And what you have, for instance, if you're interested in immunotherapy, then you want to look at the escape mechanisms of the tumor. And that's what most tumors have. They have quite a bit of escape mechanisms that the immune system cannot attack anymore. And if you look at those slides, you can see it's heterogeneous. So there are different regions with different escape mechanisms and with different strengths of escape mechanisms. So, I mean, we all know that the tumor is heterogeneous. If you do gene analysis, you already know that's really the heterogeneity is immense. So if you have that, and if you have that in mind, that actually there is also a tumor microenvironment where the neighborhood of the tumor things are happening. There's interaction happening between the for instance, the fibroblasts even, or the immune cells interacting with the tumor belongs in a way to the tumor. It's part of the tumor. It's a tumor microenvironment. And if the tumor is heterogeneous, then it's completely obvious that also the tumor microenvironment has to be very heterogeneous, because it's a kind of a co-evolution that takes place between the tumor and the tumor microenvironment. And if they co-evolve, I mean, and the tumor itself is heterogeneous, the tumor microenvironment has to be as well. And then, already here, you have a relatively complex situation in front of you. So, and what do you measure now? What kind of cells do you now do the statistics on? In which region? There are several regions in terms of tumor and tumor microenvironment. What is most prognostic and most predictive? That's not clear. And that's why the machine comes into play, because it can play through many, many different possibilities and hypotheses in this case, whereas if you don't quantify and just have a hypothesis in your mind and you test it out by counting cells and so on, it takes forever. Yeah. So the machine can do it very fast. So that's what we do when we do tissue phenomics. We just profile, and if we focus on cancer, then we profile the local tumor ecosystem. And as I said before, it's really only local. And does it still work? Also, it's so local. It would be nice to couple it to the global tumor ecosystem, learn more about that as well, and include that. And that's certainly then the next step. But for the time being, we concentrate on the local tumor ecosystem and profile this. So tissue phenomics is then about the science of discovering biomedical relevant structures, which we call phenes, like in relation to genes, in the tissue. And in particular to phenes that might not be recognized by the pathologist because it's too complex. And that's our engine for doing this. So starting on the left, we start with the images, then we do the image analysis, and then you have different slides, which are just after one after the other, so just cutting several slices, consecutive slices, and with different stains, and then you have to bring this information together by co-registering the different slices, putting them on top of each other, that all the regions match with each other, which are relevant. And then what we do, we export all the data from, for instance, the millions of nuclei, or a million, let's say, for a single slide, and you have maybe several of those stains. Let's say you have 10 stains. It's 10 million then, what you have. And then you have maybe 200 patients. So you can have 500 or more million nuclei in your system just from one clinical trial. And that's quite a big amount of data. And the question was, could you put it in a machine that can handle this, so that you can do a playful analysis with those hundreds of millions of nuclei or cells just in one data mining tool? And we found out it actually works. So you can do it. So you can do it very playfully, and in a very, very playful manner, you do statistics between in all the different regions you might think that they are important. And you can do all kinds of other statistics, not just the density of cells, but you can also look at what is the average distance between tumor cells and inflammation cells in certain regions. So all measuring relations or statistics about relations between cells. So it could be very complex, but you do it just in the data mining tool, which is then, if you test one hypothesis, is a matter of seconds, and you know the answer. So image analysis and data mining, this goes together. So what can you do with it? Prognostic is one thing. You can look whether you can predict outcome, survival, disease-free survival, recurrence. And we have one project on prostate where we have something like 150 patients with 5 years of follow-up, and we measure the PSA recurrence here in this case, and we question whether we can predict PSA recurrence just by looking at the tissue, the machine looking at the tissue. And we have also different stains, like H&E, maybe I press the button here, and CK18 and P63. So with CK18, you just mark the epithelium, and with P63, the basal membrane cells. And if on a gland, the basal membrane cells are not there anymore, that's in regions where you don't have the bright dots around the gland, like in the yellow circle you see there. There the gland is still intact, so the basal membranes are still working. Consider this as a healthy gland, but it's surrounded by many other smaller glands where you don't see the bright dots. This is cancer. And just from how that's mixed, how the healthier glands and the non-intact glands are intermixed, is already prognostic. We found that out. But what I want to talk about here more is, can we look at the immune cells and see whether they predict something here in this case. So CD3 and CD8, the effector cells is CD3. CD8 the cytotoxic cells that can kill the cancer. And then the macrophages that is CD68 is also a double stain where you have on one slide two different proteins stained to highlight the macrophages, the M1 and the M2 state of the macrophages. So macrophages can be in two different states, and it's known that the ratio of both might have a prognostic value. So we wanted to figure that out, whether that's true. So we looked first at the macrophages, and by having this nice P63 we can stain, we can look at the tumor very nicely, and it's marked on the lower left in red. That's a tumor region which is then automatically found. And then you can look at all the regions in the tumor microenvironment, which is a little bit difficult to tell for prostate. I mean, I'm not sure whether you really can talk about tumor microenvironment like for colon cancer. It's slightly different here. But still, you can look in the neighborhood of the tumor, in the tumor, and far away from the tumor, and do statistics. And now, so we have the macrophages stained, but then you have to co-register, because it's usually on the slide, it's rotated and it's shifted, and you co-register that, and then you project all the statistics you can do to the tumor regions, the neighborhood of the tumor regions, and do there the statistics. And what we found out actually is, if you now automatically split your patients into patients with a high value of some feature and low value of feature, and plot that on the Kaplan-Meier curve, you look at how the disease for those patients evolves. In this case, it's PSA recurrence. Then you can see actually, just by looking at the ratio of M1 to M2 macrophages, the density is prognostic. It's not perfect, but it's already quite nice. So you can predict just by measuring this ratio, and this ratio is here measured in the tumor microenvironment, so in the neighborhood of the tumor, and not somewhere else, and not in the tumor. And that's what the machine automatically found out by going through many different possibilities. Oh, I'm seeing that I'm running out of time. I have to hurry up a little. And then you can do the same thing with cytotoxic immune cells, the CD8, and exactly do the same thing, and also project that onto the tumor and so on. And then you can combine this with the macrophages, and you come to a relatively nice a prediction here, or prognosis. And if you look back, I have to go back, and if you look here to this Kaplan-Meier curve that was predicted through a pathologist by looking at the Gleason score. We just took the very difficult cases where the Gleason score doesn't tell you so much. And still, now, only looking at those very difficult cases, you get already quite a nice prediction for the prognosis. So more interesting is perhaps than predicting drug response. And there we went into a study, a clinical trial with, from immunotherapy. Immunotherapy is, to me, very related to nanotechnology. In principle, it's enabling the immune system to attack the tumor, and the tumor certainly develops these escape mechanisms, and you have to overcome those escape mechanisms by helping the immune system and by blocking, for instance, those escape mechanisms, by stimulating at the same time the immune system. You can do many, many different things to help this nanotechnology of nature. I mean, the immune system attacking the tumor is kind of a nanotechnology interaction of the immune system with a tumor. But now you bring in artificial nanotechnology by helping the immune system and by maybe helping also to get rid of these escape mechanisms in the tumor. And right now, I would say we are in a state where that's very early. You just bring in certain molecules that block something, or they stimulate, that bind to certain proteins on the surface of immune cells or on tumor cells. But this is not the final word, I would say. Here, I think nanotechnology can play a major role by dealing with the complexity of the immune system and the tumor. Both are immensely, I was overwhelmed. I just now working in this field since a year, and I was completely overwhelmed how complex our immune system is. And the drugs we have today on this are very simple. I think they should get more and more complex. You need to do some nanotechnologies and do some logic here. Not just having one type of molecule, but not only having combinations of molecules where people are thinking about, but having logical combinations of molecules. That's something where I see a big future for nanotechnology in immunotherapy. And immunotherapy is a very extremely promising field, because many people get healed through immunotherapy that had no chance for getting healed in the past. So then saying that, that's a typical stain of an immune cell. So we did not have much information, just two stains, tumors. A tumor escape mechanism is stained, and the immune cells are stained. For some reason, I cannot give you all the details of this investigation because it's now not our data. It's in the collaboration with MedImmune, which is part of AstraZeneca, and it's their data. So, but in the near future, I think we can publish all the details as well. So what you can then do, you can do automatic cluster analysis. So you don't tell the system what regions to look for. The machine should find it. Yeah? It plays with all the different regions, does some cluster analysis, and then spits out a result. You have no clue what the machine did, but you get a result which is not so bad. Yeah? But then you have to, in the next step, you have to figure out what it really means. What did the machine really find? That's maybe a complex procedure, and you cannot sell such a result without giving some explanation. That's what we figured out. So to the FDA, I think you have to give some explanation what it means. But you can give some more natural information to the machine, saying, yeah, just look into the tumor microenvironment or in the tumor in general. It doesn't matter whether it's heterogeneous or not. Just measure it in all the tumor and do some statistic of immune cells or something like that. And combine it maybe with a second feature. And then the machine can bind all features you have together and come up with a logic what is predictive in this case. And that's what we did here. And then the other thing is you just go and do some hypothesis-driven analysis. And you say, yeah, the tumor microenvironment, and maybe I look at only at escape mechanism regions in the tumor, and combine it with the immune system, the density of the immune system in in the tumor microenvironment and as well in the tumor itself. And if you do this, you can see here for one example where you have the immune cells, and then you have the tumor stained, the positive in terms of escape mechanism, positive tumor and the negative tumor, and you now again do the same thing, co-register, and you see where the immune cells are attacking the tumor. And from this pattern, you then can do that for all kinds of images you have in your study, and then you can do prediction and split your patients in two groups and find out, can you predict? And from the Kaplan-Meier curve, you see really the prediction is not bad. And the PPV, the positive predictive value, is here over 50%. It's not perfect. Yeah? That means from all the patients on the upper curve, which are surviving quite nicely, not all of them are actually responders. Responder is defined here in this way, that the tumor really shrinks drastically by more than 30%, at least at one stage. PPV of 55% means only 55% of the patients we predict as being responders are indeed responders. The others are not. So it's not yet perfect. But I mean, the first steps, I would say. And then we need to validate that by doing a blind test and without knowing anything about the system, and a person has not seen the clinical outcome data ever, does on a new data set a test, and it looked very nicely. So from this, we learned that this might work very nicely, and we did it on more patients. And then we found a little bit of surprise. So we got the same exactly very nice result, but it made a difference whether the patients were first-line patient or third or second-line patients, or whether they were already treated by something else beforehand, or whether they were treatment naive. Yeah. And then we figured out, we trained on third-line patients, and for third-line patients it works very nicely. And for first-line, now we have to, we came up with a new algorithm, which now works, but we have to validate again now. So it's not completely easy, but I think I can say it at least it works very nicely. It works better than other methods today. And we are just at the beginning, and I think it's very promising. Thank you very much.
Thank you very much. We have time for a few short questions, please.
Simo, you may go until 40.
Okay, great.
So there's more questions.
Okay. So anyone in the public? So let me start with a few of my questions. So I think that the most interesting use of this technology probably will be to predict response, but in particular in those cases in which patients have been treated and they are already in a kind of resistant stage. So, I mean, patients that you treat, then they become resistant, so the tumor changes, and then you need to know whether in this second line of treatment, this patient need to be treated with the same drug, or you can actually jump to the second line of treatment. So, did you have any experience in that with the system? So, trying to find out so how to predict this in particular. My question is how to predict this in those cases in which you already have surgically removed the tumor. So, can you do that by, for instance, analyzing biopsies from metastatic sites, or?
Yeah, I mean, the cases I presented, maybe, sorry, I forgot to mention that was exactly this. It was biopsies from, it was lung cancer, and it was biopsies from the lung, and as well as from metastases.
So you can predict from metastases?
Yeah, that was a very similar algorithm that has been used, and like you said, for the patients that had been treated or not had been treated. I think we can predict both in both cases, but the algorithm is different. So it's not exactly when you have to predict something for treatment-naive patient, you use a different algorithm. It was surprising to us that this is, but I mean, how you explained it, I mean, it seems for you it's obvious, I have the impression, that they behave differently.
Patrick, please.
Thank you very much for this nice contribution. Now, you are turning images into big data. You analyze them to get prognostic information. In the very first step, you select a subset of data. And that means also you throw away some information which you don't consider too important. Now, do you do this because it's just the computers of today are not powerful enough to do the computation on the whole data set? Or is there a different reason?
Yeah, I mean, you throw away the genetic information and all the patient data. That's
but all, even from the image information, if you say I go down by selecting, by making regions of interest, by looking for intact cells, that also means a selection of the
Yeah, that's right, but the selection is done in a more intelligent way. So if you have certain regions that don't tell you anything, they don't correlate at all with clinical outcome, then we throw it away. Otherwise, we don't. Yeah, so that you go through many different possibilities. But I have to admit, you cannot go through all the possibilities that are thinkable. Yeah, that would be too many. Yeah, I agree in this respect. You have to limit yourself to some extent by some hypothesis, otherwise, or some algorithm that limits your variations of possibilities. Otherwise, the machine would not be big enough, and would not calculate in the next 100 years or so. If you go through all the possibilities too much. Yeah.
And you use a supercomputer, a notebook, the new Chinese machine.
No. In this case, it's just a notebook. Yeah. The image analysis is a little bit more time-consuming. This is done on clusters, so for every patient is on a separate computer, so to speak. It's all analyzed in parallel.
Okay.
We have a previous question there, Mia.
Yes, thank you very much, and I'm happy that you raised all this analysis problematic, issue that is very debatable nowadays. And following what Patrick says, I wanted to ask you, we never have enough repetition. We never have enough, samples to really do the right way. So therefore, as you said earlier, you do this stepwise. But still, if we are talking about imaging, and since we cannot analyze all relevant or all important data or features, even if it's only the imaging features, we cannot have the genetic and the images and the clinical, and maybe we need even more at the same time. So we developed new tools like you presented some, and we do it stepwise. But do you have a feeling how much or what is the price we are still paying by doing this stepwise and not having a tool that could really tolerate the whole full one-step analysis?
Yeah, it's a combinational problem. That's why you are limited. So you have so many possibilities how to combine and how to make hypotheses automatically that this takes forever. And what I present is, in principle, not yet really big data, because, if you just look at 1,000 patients or even 100,000 patients, it's not really big data. We are on the way to big data, and we start, in a way, with small data and learn from this. I think you have to learn first, and that's why you have to do it stepwise, because while you are doing this, you learn more, and if you then, in the end, do big data, then actually you are in a better position, because then if you know better what you are doing, you are in a better position because you have a great statistical background, and then you can look into the details. If you look with small data in details, then you tend to overtrain the system.
But if I may, the big data is a big, fast buzzword, and I think we are very far from there, and we need tools to evaluate our real mistakes, our real deviation, because even if we'll have millions, just one voxel needs 30 billion, so you will never have it. So we have the tools to tolerate the mistake and to embed it. And I don't think we have them now.
Yeah, I agree, not completely, because, I mean, big data is in other domains, is already very successful. The industry uses those algorithms to predict customer behavior and how something material is ordered, in what kind of frequency, which helps them a lot to save money. They use it. So it's working to some extent. But, I mean, you need to have a little bit higher standard if you apply this to medicine. There I agree. And we are not completely there yet. So that's why I think starting with not completely big data, but smaller data, and learn from this and evolve in the direction of big data, in my opinion, is the right approach.
We have time for another question here.
This, right here. Thanks very much. And I'm going to go back and tell my pathologist to start to look for new jobs. But I'm curious about the time element, and you mentioned it slightly, because I could see some value to this in adaptive trial design when you're going through clinical trials. But can you say a little bit more about how long does it take to get the answer? That whether it potentially will have the ability to impact in an adaptive trial design, for instance? Yeah,
actually, it's, as I said before, if you have all the data in a data mining tool from all these objects that are I mean, hundreds of millions, and you still can analyze them in one machine, even on a laptop, then actually you're really fast. So the answer you get is a day, let's say. If you're going through many iterations, is a day. That's roughly the time scale. You have not tried everything. You restricted yourself to some hypotheses, but you went playful through many different options, and still, in some cases, if it's even more limited, you get the. If you just want to test one hypothesis, you know the answer in a second or in some seconds. Yeah.
Okay, thank you very much. It's lunchtime, and I know that Beat is hungry. He's making me some strange signs. So I'm sorry, but I know that you have a lot of questions. I'm sure that you will have time during the lunch break to proceed. Thanks very much for coming.